Okay, let's dive right in. We're about to explore the absolutely spectacular, sprawling, and let's be honest, sometimes really strange world of kingdom Animalia. We're going to unpack the fundamental blueprints, the basic designs that bring some sense of order to the incredible diversity of animal life on our planet. So, here's the big question, right? When you look at an animal as simple as a sea sponge, and then you look at something as intricate and complex as a spider, how in the world do biologists even begin to tell them apart? What are the rules? How do they sort through millions of species? Well, that is exactly what we're here to figure out. And the secret, it really lies in something called the body plan. The best way to think of it is as the fundamental architectural blueprint that every single animal is built on. And once you start comparing these blueprints, you start to see this beautiful, logical order emerge from what looks like total chaos. And believe me, we need a system. Scientists estimate there are somewhere around 9 to 10 million different animal species on Earth. That is just a staggering number. And this idea of a body plan is our #1 tool for making sense of it all. So where do we even begin with all that diversity? Well, the first major dividing line in sorting all animals comes down to their basic shape. It's all about something called symmetry. So what is symmetry really? It's just the way an animal's body parts are arranged. Can you draw a line right down the middle and get two matching halves? Or maybe you can slice it multiple ways, like a pizza, and still get equal pieces. This simple idea, this property, tells us a surprising amount about how an animal actually lives. Let's start with the simplest forms. Over here, you've got the sponge, which is asymmetrical. There's just no way you can cut it to get equal halves. But then you have the sea anemone, which has what's called radial symmetry. You can slice it right through the center from a bunch of different angles and always get mirror images, just like a pie. And this is actually perfect for an animal that stays in one spot, because it can sense food or danger from any direction. And that brings us to the next massive leap forward: bilateral symmetry. With this, there's only one way you can slice it to get two matching halves. It creates a distinct left and right side and a front and a back. This whole arrangement is what allows for the development of a head, a process called cephalization, which leads to purposeful forward movement. I mean, having a designated front end with a brain and sensors was an absolute evolutionary game changer. This table really breaks down the key differences for us. You see, radially symmetric animals, like a jellyfish, they pretty much never have a head. But animals with bilateral symmetry, that's us, insects, fish, you name it, almost always do. That development of a head is one of the biggest features that separates the vast majority of animals from these simpler forms. Okay, so once we've sorted animals by their external shape, we've got to look deeper. The next level of classification is all about the internal architecture. How many tissue layers they have and what kind of body cavity, if any, forms on the inside. The earliest animals were what we call diploblastic. That just means they had two germ layers, an outer one, the ectoderm, and an inner one, the endoderm. Jellyfish are a great example. But the huge evolutionary leap was the arrival of triploblastic animals. They have a third layer, the mesoderm, sandwiched right in the middle. And this mesoderm, this is the key. It's what gives rise to things like true muscles, bones, and all sorts of complex organs. Now, inside that all-important middle layer, a space can form, a body cavity. But it's so much more than just empty space. So, what is it actually for? And why is it such a critical feature for telling different animal groups apart? Well, it turns out there are three main options here. You've got acoelomates, like flatworms, which are just solid tissue all the way through. Then there are pseudocoelomates, like roundworms, which have a kind of false cavity that isn't fully lined by that middle tissue. And then you have the coelomates, that's us, by the way, with a true coelom, which is a fluid-filled space completely surrounded by that mesoderm layer. So what's the big deal about having a true coelom? Well, for one, it acts like a protective cushion for your internal organs, letting them grow and move around without being squished by your body wall. Plus, that fluid-filled space can act as a hydrostatic skeleton, which allows for way more complex movements and body shapes. It's a huge advantage. All right, we've covered shape and internal structure. The next great divide in the animal kingdom is one of the most fascinating and, frankly, one of the weirdest. It all comes down to what happens when an animal is just a tiny little ball of cells in an embryo. So check this out. As an embryo develops, a little indentation forms. It's called the blastopore. And the fate of this single opening determines which of two massive supergroups an animal belongs to. In protostomes, which literally means mouth first, that little opening becomes the mouth. But in deuterostomes, or mouth second, that opening becomes the anus, and the mouth forms completely separately later on. And yeah, it really is that simple. This one single developmental event separates animals like insects, spiders, and snails from starfish, sea urchins, and us. It is a fundamental split in the tree of life that happened hundreds of millions of years ago. So now that we have our rules, we're looking at symmetry, layers, cavities, and embryonic development. Let's see how they apply by looking at the undisputed champions of kingdom Animalia. Meet phylum Arthropoda. We're talking insects, spiders, crabs, centipedes. These creatures are absolutely everywhere. Their basic body plan is, without a doubt, the most evolutionarily successful blueprint on the entire planet. So what's their secret? What makes them so successful? Well, it really boils down to three key innovations. First, a hard protective exoskeleton. That's like a suit of armor. Second, their bodies are built from a series of repeating segments, which allows different parts of the body to become specialized for different jobs. And third, those incredible jointed legs and appendages that act like levers for all kinds of precise and powerful movements. And this one basic blueprint was just so adaptable, it was modified for total world domination. You've got insects that took to the air, crustaceans that conquered the water, and arachnids and myriapods who became masters of the land. It's an incredible story of success, all stemming from one winning design. So when you step back and look at the big picture, you see that these rules, symmetry, layers, cavities, development, they're not just random boxes for scientists to check. They are clues. They let us trace the evolutionary history of life and reveal the hidden order, the hidden connections between all animals. Which brings us to a final, kind of fun question. Based on everything we've just talked about, what fundamental trait do you share with a starfish that you do not share with a spider? And the answer is pretty surprising. You are both deuterostomes. Back when you were just a tiny embryo, that very first opening that formed took the exact same path in both of you. It's a deep, ancient connection that actually places us evolutionarily closer to a starfish than to a spider. Wild, right? Ultimately, these fundamental blueprints are what define the shape of the tree of life. They reveal these incredible relationships we would never guess just by looking at an animal from the outside. And it really makes you wonder, what other profound, unseen connections are still out there, just waiting to be discovered?